Dr. Alejandro Sanchez Alvarado is currently an investigator at the Howard Hughes Medical Institute and the Stowers Institute for Medical Research. He's also a fellow of the American Academy of Arts and Sciences and a fellow at the Marine Biological Laboratory in Woods Hole, Massachusetts. His undergraduate degree is from Vanderbilt University, and he attended the University of Cincinnati College of Medicine for his PhD in pharmacology and cell biophysics. His research focuses on regeneration, and I learned last night from him that cells are evidently more decisive than I am, and that I need to learn more about epigenetics. I met Alejandro many years ago, when we both lived in McTier International House at Vanderbilt University. To be honest, at that time, I didn't even track that you studied biology. I knew Alejandro as someone who loved languages and culture and music and philosophy and liked to stay up late and think big thoughts and talk about them. But as you're about to find out, he's most definitely a scientist, and we are so pleased to have him here. Please join me in welcoming him.
Thank you, Jen. Thank you. Thank you. Thank you very much for that very kind introduction, and thank you very much for coming. And I'm going to start by starting at the end. And by that, what I mean is that I'm going to start at the end of the life of this particular painter right here, which is Diego Velasquez. You can see him right there. This painting, I think, is a very strong representation of why we really need to think big and think hard about where is it that we're going to go and what we're going to do with our lives. And why do I say this? The painting was painted in 1656. 4 years later, Velasquez would die. And so I believe that in this particular canvas, Velasquez was really trying to collect the sum total of his expertise, his talent, his genius, and his intellect, such that it could remain as evidence of a life well lived, right? And so what I like about this painting is that it's literally a snapshot. It's something that will be very familiar to us today when we pull out our phone, take a picture, and you just freeze in time a particular moment of your experience. And the reason why I think it's a snapshot is because essentially almost all of the people in this painting are looking at you as if you had just walked into the room to see what they were doing. And that's in fact what happened here. So you have a situation where this individual right here is actually looking at you. So is one of the chaperones, so you can barely see right here. This guy by the threshold of the light back here is also looking at you. The painter is looking at you. And the princess, Teresita right here, only the eyes have moved to look at you. She hasn't even turned her head yet. This is a beautiful painting. And then what you see about this is that in order for this painting to be as powerful as it is, Diego Velasquez not only needed to know how to paint, he needed to understand geometry. He needed to understand sciences. And this is one of the beautiful things about this work of art, is that when you dissect it and you look at it closely, what you see essentially is that there are two paintings up there. They're from Paul Rubens, and they are as a pair. You also see that the individuals in this painting are distributed in a very symmetric way and repetitive way. You can see, for example, that these three individuals are right in between the columns that separate the painting right here, this mirror, which we're going to talk about in just a second, and this other threshold. You can see this lady here, the consort, and this one over here, and of course, the princess right there. Now, what you're seeing here in the mirror is the people that just walked into that room. Those are the Queen and King of Spain, Philip IV and his wife. And essentially, everybody now is looking at the Queen and King coming to visit the studio of the painter. So this painting actually allows you to sit or stand on the shoes of royalty. You are now the king and queen walking into his studio. And what is really cool about that is that all of this harmony is essentially controlled by the vanishing point, which is essentially the hand of this individual right here, forcing you to essentially look at this particular area. This is a beautiful piece of art, and I think it's a vigorous argument for the virtues of not just art, but also science. So, if you continue to look at the Western canon, math and sciences will actually pop up in the most unexpected places. And so this is one of the things that I like to study. I like to study scale and proportion. Why are animals and plants as big as they are? We don't understand. why humans are as tall as we are, why ants are as small as they are, why whales, for example, are as large as they are. And so in this particular book, that's what Jonathan Swift in Gulliver in the Land of Lilliputians was actually addressing. Gulliver was a regular-sized human being that just happens to land in an island where all human beings are really tiny. They're called Lilliputians. And I didn't realize that he had actually done some math until I was reading this book to my son when he was younger and realized he was actually doing Euclidean geometry. And so here's what he writes. He wanted, the king of Lilliput wanted to understand or know how much food should we feed this giant. Because if he gets hungry, he might eat all of us. So we need to make sure that we're going to feed this guy. So he called the scientists of his court and said, please calculate, what we need to feed this human being. And so here it is. So it says right here that the conclusion was that the emperor received the following numbers. 1,728 is the number of rations that Gulliver must eat. And I thought that's a pretty random number until you actually look at the rest of the text. So the way they did this is that the scientists went and they measured the height of Gulliver's body. And then what they did essentially after measuring the height, they realized that it exceeded theirs in the proportion of 12 to 1. And so Gulliver's body must contain at least 1,728 of theirs. That's essentially just Euclidean geometry, which is 12 to the cube, which is 1728. Immediately you realize that this is not metric. This was written in the British Empire. They're using British units of measurement for feet and inches, and that's why it's to the cube, right? And so that was the Euclidean geometry they did. So if you take the 1/3 of 1728, that's 12. So they concluded that they needed to feed enough food for this giant that was equivalent to 1,728 of theirs to keep him happy. And that's one meal, okay? I don't know how many little sheep he had to eat that when he was there. But this is the problem, is that, the science stops there because food intake and biomass, they do not follow the same scaling component. Euclidean geometry is something we invented. It's not something that really occurs in nature. It's a way that we as human beings invented to interpret volume and space and distributions of those. And so it turns out that the way scale and proportion are regulated in humans actually follow a very different equation, which explains it or describes it, but does not explain why we scale the way we do. It's called the allometric equation. And I'm not going to tell you about all of the background that went into the discovery of this equation, but essentially what I want to tell you from this is that this component, or this particular equation right here, really explains how much food you need to eat in order to keep your biomass. And you'll see is that it is not to the third power, but it's 3/4 of a power. And so, in fact, what Gulliver must have eaten instead of 1,728 rations should have been 268. So he gained weight. Okay, but that's not reported. That's not reported. He must have gained weight. So, you can see how, for many, many years, the sciences and the arts have actually gone hand in hand as a way to interpret and understand the world we live in. So the scale and proportion issue is very important because we always start as a microscopic cell. And so this microscopic cell will actually undergo a series of cell divisions, which you're seeing right here for a fish embryo, that ultimately will produce something that we'll recognize as a fish. Okay, so you're going to see, for example, here the head forming, and you're going to see how the trunk begins to form right there. You can see what the musculature is going to be like. This is going to be the tail of the fish right here, and ultimately it's going to produce something that's more or less going to look like a fish. The reason why this is so fascinating is because we know when it starts. It starts when fertilization takes place. When you take an oocyte and you take a spermatozoid, you bring them together, the cells begin to exchange DNA information, and then all this particularly beautiful set of choreographic cell dances unfolds right before your eyes, and to produce something as complex and as gorgeous and as delicious as a fish. That's for development or embryonic development. But the kind of things that I really want to understand are the things that happen beyond development. The things that will most likely happen to all of us because we spend more time not being an embryo, but being an adult or a growing individual for the rest of our lives. And that's when we're most exposed to the elements. That's when things are much higher or having a much higher chance of causing some problems to our well-being. So I'm just going to illustrate what I mean by that by using this particular photograph, which I mooched from the internet. It's a sea star. And what I'm going to do is that using the magic of Photoshop, I'm going to amputate each and every one of the limbs of this particular organism. So I've done that. And as you can see, each of the surfaces of amputation actually have different geometry. And that's because the animal does not have any control on how it's going to get injured. This is the vicissitudes of living in the outside world. You know, if a fish is hungry and you're trying to run away and it bits you, it may have actually produced a bite that was perfectly perpendicular, for example, to the axis of the arm, or it could have been a diagonal, or it could be a jagged bite, or it could be a semicircular bite. And the remarkable thing is that irrespective of how it starts, the end product results being the same, which is the restoration of the missing body parts. So the beginning of regeneration is completely ad hoc. So just as embryogenesis had a very well-defined beginning, the beginning of regeneration is ad hoc. So we don't understand even how that is possible. Okay? So regeneration as a biological attribute is remarkably enough broadly but unevenly distributed across the animal kingdom. So you find that things like anemones, for example, can be sliced in a number of ways, and each of those fragments will go on to regenerate a complete anemone. You can see things like octopuses, for example. Their tentacles can be amputated, and they'll actually grow back again, and it's a fairly large animal. Snails, you can decapitate them, and their heads with their brain and their eyes and everything will actually grow back again. This is nature. These are not mutations. These are things that happen normally in the wild. And so these are relatively large animals capable of doing some truly remarkable things through mechanisms that we don't understand at all. And that's what we want to understand. Here's a close relative to our lineage of the so-called deuterostomes, or the branch of animals that ultimately produce or to which we belong, right? This is a sea cucumber. This is probably the prettiest sea cucumber you'll see because most of them look like a piece of turd sitting at the bottom of the ocean. It's the truth. And the first time I saw one, I thought I was swimming in the wrong part of the beach. But it turned out that they were really just a sea cucumber. So why am I showing you this? Because these organisms, when threatened by a predator, they'll actually take their entire digestive system and throw it up. They'll throw up their esophagus, their stomach, their intestines, such that the predator can eat that and the animal can slowly retreat, and after a couple of weeks, they'll regrow all that tissue again. Just think about that for a minute. They'll have to survive for a couple of weeks without eating because they don't have a digestive system, but somehow they manage to do this. How do they do this? We don't know. All right? There are animals that are vertebrates like ourselves, such as salamanders, in this case the Mexican axolotl. These animals are truly remarkable. These organisms can lose a limb and it'll actually grow it back. You can cut 3/4 of their heart and it'll actually grow back. You can remove part of their kidney, it'll grow back. You can even remove the lower, the jaw, for example, and it'll actually grow back as well. And that is a vertebrate, not too dissimilar from you and I. And yet, we don't know how they actually do this stuff. Now, normally, when I present this data to my colleagues in the sciences, they will come back to me and argue, well, you know, the reason why this works in these animals and it doesn't work in us is because all of these animals are aquatic, they secrete, you know, prodigious amounts of mucus, and we're nothing like them. And so that's why regeneration takes place in these animals. Until, of course, I show them something like this, which are the so-called ungulates, or deer, or moose. These animals, the male of the species, will actually go through, if you're a hunter, you know this, they'll actually go through this process of shedding their antlers. And every spring, they'll be growing new antlers, and that's how you set up the pecking order to keep your standing in the group of deer. What is remarkable about this is that it's incredibly fast growth, and if you look at the antlers themselves, you can see that they have these very specific looking points. Now, if you follow an individual deer through its life, and you collect the antlers every year, and you were to put them side by side, what you'll see is that the antler that's produced the next year will have recapitulated the same geometry from the year before. And so you can actually see them grow through time, even though they're separated by the shedding processes. So how do they remember? Why don't they just produce random antlers? But they don't do that. And so that means that this particular process is under strict genetic and cellular regulation. And these tissues are not just bone that grows. I mean, this tissue, when it grows, if you've seen them, they are epithelialized, which means they have a skin. They're vascularized, so there are blood vessels in there. They're even neuralized. There are neurons growing into this thing. And then somehow, all of these soft tissues are programmed to die. And so death occurs to these tissues, and what's left behind is the bony structure, which is the antler of the organism. And this is a vertebrate, a mammal. something really, really close to us, and they can do that. And their genome is not quite different from ours, and somehow they figure a way to activate these programs to allow for the regeneration, the physiological regeneration of these antlers year after year. So if you look at a tree of life of all of these metazoans or animals, everything that is in green for all these 29 phyla have animals that can regenerate. The ones that are in black, they either don't regenerate or nobody has tested them. So it suggests to me that regeneration is not something that occurs in just a very small group of animals, but really is broadly distributed across the entire collection of animals that populate our planet. And we don't know to what extent these processes are the same or different from each other. That's how poor our understanding of these processes truly is. So how long have we known about regeneration? We know about regeneration for an incredibly long time, even from the times of Aristotle. In fact, Aristotle wrote about this in his History of Animals, where he wrote that the tails of lizards, if they be cut off, will grow again. We knew this, and they already had in their own mythology Prometheus regenerating its liver or his liver, and a few other things, the Hydra being decapitated and regenerating heads. So this was already part of the Western canon as early as Greek antiquity. And so the problem's been around for, over 1,000 years, and we're no closer from solving it than Aristotle was, in 300 or so BC. So it is a very difficult problem. And so one way to solve a complex problem is to reduce it to a simpler problem that can or has already been solved. This is how engineering problems are resolved. So I'm going to illustrate what I mean by that by resorting to our friend Pythagoras. And I know that this is the one thing you wanted to do today was to do geometry this early in the morning, but we're going to do it. We're going to do it. And the reason we're going to do it is because I want to illustrate the way in which we're now beginning to think about solving these very complex problems in biology, which is not an engineering problem, it's more of a creative process by which to reinterrogate these questions again. So here's the famous Pythagorean theorem. You have a right triangle with a right angle right here and a hypotenuse C and sides A and B, okay? And then Pythagoras realized way back then, even before Aristotle, that these things were related to each other by the following equation, that the square of the hypotenuse equal the square of each of the sides added together, okay? That's the famous Pythagorean equation. There are at least 3,000 different solutions to this theorem. I'm not making this stuff up. People have been working on solving this theorem for an incredibly long time. And very diverse group of people. I mean, in fact, one of my favorite solutions was actually made by, devised by James Garland when he was a congressman before he became president of the United States. I don't know what congressmen are doing today, but they're not doing this. Okay? But this is what he did. This is his solution as he presented it, in a scientific paper, showing that, in fact, this theorem can be explained in the following equation. And then you have very complex solutions to this problem that require a wealth of pre-existing knowledge in order for you to derive that solution. And that requires you knowing algebra, geometry, trigonometry, and all kinds of other mathematical disciplines to be able to derive something like this. Well, what if I were to tell you that there's a solution that requires no pre-existing knowledge of this body of scientific research, and that you can actually produce a solution just by thinking about it differently? And I'm going to give you an example of that solution. This is perhaps my most favorite solution of this theorem. It requires no math. So essentially, you take this theorem literally. So you're going to draw a square on each of the sides of this right triangle as follows: square A, square B, and square C. Now, square C is made out of hypotenuses from that triangle, so you can decorate that square with hypotenuses and 3 other triangles like so. Now, if these relationships are true, I can take those 4 new triangles and square C out of the equation like so, and now I can do the following. I'll bring the hypotenuses of 2 of these triangles together because they are the same, they're C. If you do that, they match, no problem. And if the relationship is true, the space that is left behind should be square A and square B. And I solved it. And I didn't know to know any trigonometry or algebra or anything of the sort. I just had to think about it, right? And so this is perhaps one of my most favorite solutions to this problem because it illustrates that even complex problems, even if they have been worked out for a very, very long time, can actually be solved by thinking about them and by checking your premises and coming up with different ways of trying to get to the crux of the matter. And so that's what we want to do with regeneration. So what I thought we would do about 20 years ago when we started working on this was, can we find a simpler system? And can we learn from that simpler system, I guess, fundamental principles or first principles that will allow us to get to the matter of regeneration? I want you to bear with me for a few minutes because I'm going to give you a little a metaphor of how to think about this. Just imagine that we just invented a time machine and that we're able to bring Leonardo da Vinci to the present time. And Leonardo da Vinci wanted to fly. This is one of the things that he was most fascinated by. And you can see he made drawings for these things. He even made a drawing for a helicopter. Clearly he was fascinated by flight. And now we can tell, hey, Leo, we can fly. We have machines that actually are heavier than air and they can fly. And we show him, our pride, Boeing 747 coming out from Seattle. It's a gorgeous machine, really remarkable. And we can also show him another machine. This is a Spitfire from the UK that essentially stopped the Germans. I mean, you'll see that's miraculous that we're able to do that during World War II. So, but both of these machines, are very different from each other, but both of them do the same thing, which is flight. Now, if we bring Leonardo da Vinci to the cockpit of a 747, this is the first thing that he would see. And you remember that Vitruvian man of the guy, he drew, showing bilateral symmetry. The first thing he would see is like, oh, look, it's bilateral symmetric. It's a mirror image. You can draw a line in the middle. There's two seats. All controls are redundant. There's all kinds of similarities between both sides of this. And so now we start explaining to him how this works. Well, these particular pieces of machinery are complex, and they have, over 16,000 computer chips that control everything from cabin temperature and pressure to fuel mixture and navigation equipment, okay? And the other thing that becomes apparent is that all of these major systems have double or triple backups, things that we call in biology as redundancies. So if the landing gear doesn't drop, there's a second way to drop the landing gears. If that doesn't work, there's a third way to drop the landing gear. So there's all these built-in redundancies. And then we go and take him to the cockpit of a Spitfire. And what you see here immediately is that this was actually made with glue and rubber strings. It's not something that was really well built because there was not a lot of material in the UK at the time to build these things. And so the first thing that you notice is that there's no bilateral symmetry, nothing. I mean, there's only one handle in the middle. It's much simpler in structure. There's no computer chips. All major systems have no backups of redundancies. You lose the landing gear, you lost the landing gear. And so, if you really want to understand how each of these machines flies, I think Leonardo would have chosen to study the simpler machine. Why? Because identifying key components in a simpler machine, when removed from it, will tell you immediately what may have gone wrong in that particular machine. So let's say, for example, that Leonardo wanted to understand what a little knob did in a 747. And he goes in and removes it, asks the pilot to take the plane on flight and lands it, no problem. And so he learned nothing from removing that particular component. If you now take the same equivalent piece of equipment from the Spitfire, the pilot will say, I'm not going to fly that thing because I will not be able to deploy the landing gear. That's the button that deploys the landing gear. So now he learned that that button was responsible for that process. So in biology, we like to think that that's what we're going to do. And so this is just essentially what I just told you. I just want to get back to this, okay? Sorry, let me go back. Yeah, sorry, I'm missing that. Anyways, so what I'm saying is the following, is that you have a situation where if you want to understand how a part of our body works, we have a lot of redundancies. In fact, our bodies are much more complex than a 747 by a lot. I mean, by a lot. So if you remove one neuron from the brain, you might not learn what that neuron is doing. But if you find a simpler system that only has like 100 neurons, for example, like a nematode, if you remove that neuron, you'll immediately find out what that neuron is doing. So we need to identify simpler systems to carry out this process. I'm just going to tell you an idea why this is really the same problem. So the critics in the audience would say, well, flight obeys the same physical rules. There's lift, there is aerodynamics, all of the things are important. And we have no idea if, say, for example, a different organism does a biological process in a similar or identical way by which humans do it in the same process. And I'm going to tell you that this is definitely not true. So what you're looking at here is an electron micrograph of yeast. This is the fungi that gives us wonderful things like bread, beer, and wine. So I really like this organism. But what we know is that we can do genetics with this thing, and we can actually go in and remove genes. just like Leonardo would by removing little pieces from the Spitfire, and know what those genes are doing. And so we learned that if you remove certain genes, the yeast won't be able to divide, or they will not be able to produce specific enzymatic things, like, for example, produce alcohol or CO2 for making bread. So if we delete some of those genes, I mean, it's actually lethal for the yeast, and they die. So when genome sequencing became possible, and we began to sequence the genomes of humans, yeast, and other organisms, what became readily apparent is that a lot of the genes that we found in yeast were also present in humans. And people began to do the following experiment, which is, if I take the human gene that codifies for a protein that is absolutely required for yeast to survive, and I take that gene and put it into a yeast where that gene has been removed, would it work? And so you take the DNA from humans, put it back into yeast, and the bottom line is that, yes, you can actually rescue the lethality of yeast by replacing their lost or damaged gene with a human gene. And this is not just one gene. In fact, there are at least over 100 human disease genes that can complement the corresponding yeast gene mutation. That argues for really deep conservation of function. So it's not that dissimilar from, the airplane analogy that I just shared with you. So these genes are essentially doing the same work. So it's much easier to understand the function of that gene in yeast than it would be to do the same work in humans. Okay? So I'm going to give you an example. You can barely see it, but this is the organism that we use to study regeneration. What you see here is the head of the planarian with the 2 photoreceptors right here. You can see this tube coming out, and that tube is actually ingesting food, and that's how they eat. And these animals are cute. They look like cartoon characters, and they're hypnotic. You can look at them under the microscope, and you can spend hours looking at them. They glide like ice skaters on a petri dish. So here's a better picture of it. You can see the tube of the pharynx coming out. These animals are simple, but they also have complex organ systems. So they have, for example, a gut, which is illustrated by these white lines right here at the top and at the bottom of the animal. They also have a central nervous system that is much smaller than ours, obviously, but contains essentially all the neuron types that you and I have. They have dopaminergic, cholinergic, serotonergic neurons, etc. And here's the tube of the pharynx that we can visualize. And the reason why I'm showing you all this is because in a single picture, we can actually look at all these 3 organs under the microscope simultaneously. We can't put a human being under a microscope. It's just not possible. But now you can see the sum total of all of the components overlaid on top of each other, and now you can see what relationships may or may not exist between all these different components, and learn from that how form and function arises, particularly during regeneration. And so we can also measure gene expression, so we can actually see what cells are turning on what genes. So each and every one of those little purple dots, they represent a gene being turned on in that time, in that cell, in that particular location of the animal. We can actually look at all of them and quantify every single one of them. We can't do that with humans. In fact, we don't even know how many cells we have. Just think about it for a minute. So if you've been paying attention to the news, there's this human atlas, human cell atlas enterprises being now executed across the globe, such that we can finally learn how many cells exist in the human body. These animals have maybe between 50,000 to 60,000 cells, and we can count every single one of them. All right? And so we can do this by looking at their gene expression. And they're cute. They look like little ghosts. Okay? So this is what the animal looks like, and this is how they reproduce. This is where things are going to start to get weird. Okay? And so what you can see here, they love to do this at night. Here's an animal that began to stretch. Here's the head of the animal. Here's the tail of the animal, which is anchored on the substrate on which the animal resides. And what you're going to see in just a couple of seconds is that this is actually going to pinch off. And there it goes. Yes. So what happens here is that the tail, which is now left behind from the rest of the animal, will do the following. It'll grow a new head. And the head that lost its tail, it'll grow a new tail. So the way they reproduce is by cutting themselves in half, pretty much. That's not, don't do that at home, okay? But this is how these guys do it, okay? So this is already a naturally occurring, solution to the problem of their reproduction, something that happened in nature on its own, okay? You can also help them by using a razor blade, which you can see right here. And you can see that each and every one of these fragments is produced by a blade. But this Scotsman, J.G. Dalyell, actually called them immortal under the edge of the knife. And why did he call them that? He called them that because each and every one of these fragments, in a very short period of time, within a week, will actually go on to produce a complete animal. So you can cut these animals into 3 pieces, and each of those 3 pieces will actually produce complete animals. So this is not too dissimilar from imagining me cutting my little finger and watching that little finger regenerate me as a person completely. That's what these animals are doing. And so you can see the trunk here, and you can see the tail here doing the same thing. And these little dots right here, in these animals right here, those are the cells that are actually beginning to proliferate in order to produce these new structures, like the new head, the new tail, and the new head and tail in these 2 other fragments right here. So it is a really beautifully coordinated choreography of gene expression, cell division, and morphogenesis that ultimately produce something that looks recognizably as a head or as a new organism, as a new planarian. You can do crazier things, like for example in this experiment where we actually cut the animal into 18 different fragments, okay? And each and every one of those little fragments will actually go on to produce a complete animal. That's 18 animals for the price of 1. So I love it because it's cheap. But what this means essentially is that fragments that had never had the job of making a head need to make a head now. It had never made a head before, but it somehow knows how to actually build a new head from scratch. So this is really massive, massive organogenesis, which is something we really don't understand. So we want to exploit this biology, and we want to exploit this naturally occurring exaggeration of regeneration to try to understand what molecules are responsible for these processes. So I'm just going to show you one example of work from our lab from a few years ago, where we actually discovered a molecule that's very important in telling the cells whether they're going to make a head or they're going to make a tail. So the experiment is very simple. We take an animal, we cut it into 3 fragments, and I showed you this before. Each of those fragments will go on to regenerate completely well-formed organisms. So there's 3 animals right there. Right? If we now look at this molecule, this is the crystal structure of a human molecule known as beta-catenin. Beta-catenin is a molecule that's involved in colon cancer. All right? And when this molecule goes bad and the signaling goes bad, you actually get tumors in your colon. It's usually a very lethal disease. And so planarians actually have beta-catenin, but they don't have a colon. Let's just put it that way. But they have beta-catenin. It's almost indistinguishable by nucleotide sequence and by amino acid sequence to each other, so we know they are the same proteins. So we thought, well, why don't we eliminate this protein? Just let's do the Leonardo experiment. We're going to eliminate this protein, and we're going to ask what happens to planarians during regeneration when you eliminate this protein. So we get rid of the protein, and then we do the same experiment. We cut the animals into 3 fragments, and then we follow the fragments. And what we get is this. We get animals that instead of getting a head and a tail, they now get heads at both ends. This is like Dr. Dolittle, pull me, pull you, right? But that's just the elimination of a single protein resulted in an animal that now is only going to make heads. So then we ask the following question. What happens if, instead of eliminating the protein, we increase the concentration of this protein in the cells to see what consequences it might have during regeneration? So we do that experiment. We cut the animal into fragments, and then we look at the fragments. Now what we get is this, that an excess of this protein results in the production of tails at both ends of the animal. It's almost binary. This molecule is a switch that allows cells to decide whether or not they're going to make a head or to make a tail. And so this was really unexpected, and it tells us something about how little we actually know about the normal functions of proteins in the absence of disease. Remember, I actually mentioned to you the word disease genes, right? I mean, I don't see why we would select for disease genes. This is really a construct of our own pathetic thinking about how biology actually operates. But then there's another experiment, and this is the one that I convinced my graduate student at the time to do, which is the following. Let's just get rid of the protein, but let's not cut the animal. Let's just see if it's doing something in the absence of regeneration. And when we do that, this is what we get. We get animals that now begin to grow heads everywhere. Right? So what that means essentially is that this stable outward appearance that we actually give to the world is actively maintained. You're not done. I mean, in order for you to be able to recognize yourself the next morning, this is an active process. Things are being removed and they're being rebuilt throughout the duration of your life. And that's what this experiment is telling us, is that to maintain the proper polarity and the proper structure of the animals, these systems are constantly active. And so I wonder why we're not dropping dead more often than we are. A lot of things could go wrong, and they're not going wrong. That's how robust the system is. So I find this quite astonishing. Okay, so why in these animals? I mean, why do we want to do this? Well, I mentioned to you that there's this similarity in sequences. So when we look at the human cells and we look at the planarian cells, what we find is that they share a lot of morphological attributes. We find that their stem cells are actually very similar in morphology to each other, and that these genes that are present in the DNA are actually quite shared. These sequences are very similar to each other. So I think that we can learn a lot from looking at these simpler systems in order to understand a little bit more about our biology. And so why is it that we're so bent in thinking that we are, superior to all these other model systems, and that we're only going to use these model systems, to help us understand our own biology? I think that's really Aristotelian and sociological construct. But what I'm showing you here is a map of relationships of thousands of species whose genomes have been sequenced, and showing how they are related to each other. It's almost like a genealogical tree of the family of life. So represented in this tree, you see plants, you see protists, you see bacteria and archaea, you see fungi, people love fungi, and then, of course, animals, okay? And so you're wondering, well, and this tree essentially represents history of life on this planet. So this is a single slide to represent about 3.5 billion years, okay, of life in this planet, which is the power of being able to visualize complex ideas this way. Now, what we ask is, where are humans? We are right there. I mean, in like nowhere, okay? We're surrounded by mice and some other, parasite. This is really an unremarkable position. Nature doesn't really care that we are thinking individuals, that we are probably the only species that can actually do experiments. It doesn't care. So I think that one distinctive attribute of human beings in comparison to all these other species is that we really are the only species that can actually do experiments. And I don't care if you call those a painting or you call that a laboratory experiments. They are all experiments in trying to understand the universe around us. So I think that there's a really good reason for us to actually ask those questions. There's no other species that will ask that. And I don't know of a better way to understand life than to doing experiments ourselves. Okay? So that being said, why do I think that we are actually really just scratching the surface of all the biological space that we need to study in order to understand our biology even better? So this will bring me to this particular situation, which is an interesting coincidence. What you're seeing here is a star, a map of stars, of the constellation Cepheus, okay? And there's a star right here called Gamma Cepheus right here. And this was the first time that somebody was able to identify what appeared to be an extrasolar planet. First time, okay? Now I can understand why it would take us, over 500 years to discover an extrasolar planet. This stuff is not close. It's about 12 parsecs from us. And so all they were able to see was that with some frequency, when you aim the telescope to that star, you would see a little blip of darkness and it will disappear. And there was a frequency to it. And they argue that that's because that's when the planet was between the star and the telescope. So it was actually rotating around the star. It was controversial in 1988. And that's fine. Now we know there are thousands of exoplanets out there because we began to look. And we're looking at things that are light years away from us. I want to show you a different type of constellation, which is shown right here. It's a bunch of little microscopic organisms. This is an algae known as Prochlorococcus Marinus. This thing is likely the most abundant organism on Earth. You can actually see them, okay, from the space station. There's like fields of green on the ocean. That's Prochlorococcus Marinus. And you'd think that by now, we bathe in that stuff. We probably swallowed it when we went to the beach, okay? But these animals, or these plants actually, were also discovered in 1988. We're not looking. I mean, we're spending a lot of time looking outside. We're not spending any time looking at our own house. And so this is actually remarkable that, that there are so many things about biology, so many things about our planet that we just really don't understand. And the rate at which we're destroying it makes me really, really stressful because there's a lot of things that are disappearing that we need to understand that we'll never know existed. And we'll probably save a lot of things. So I want to conclude with the following quote. And this is a quote from a science fiction writer, Robert Heinlein, which reads as follows: that a human being should be able to change a diaper, plan an invasion, butcher a hog, con a ship, design a building, write a sonnet, balance accounts, build a wall, set a bone, comfort the dying, take orders, give orders, cooperate, act alone, solve equations, analyze a new problem, pitch manure, why not, program a computer, cook a tasty meal, fight efficiently, die gallantly. And specialization is for insects. And that's what's happening to us. We're becoming highly specialized, and we're beginning to lose sight of the larger picture, the actual masterwork that every one of us should actually execute with all the training and education that we're doing. So I'm just going to end where we started, which is with this picture. This is really a masterwork of an individual that dedicated himself to the pursuit, a singular pursuit of a single thing, and he really tried to do the best that he could. And so I'm hoping that, as you embark on your education, that you will take all of that information, and that the masterwork you produce, when you get as old as I am, I'm not quite there yet, but when you're as old as I am, is that something that you really have been honing and cultivating as vigorously and as disciplined as possible. And with that, I'll conclude, and I'll take any questions if you have any. Thanks very much.